برزنتيشن افتح هذا يفتح يمه.
ايه بس انا اشوف الريكورد مالنا يسجل شوف الصوت.
وين شو الطلاب شلون ما حد داخل. السلام عليكم ورحمة الله وبركاته طلاب الأعزاء. هاي المرحلة المحاضرة الثانية للمرحلة الثانية كلية طب الأسنان للسنة الدراسية 2020-2021. بالمحاضرة السابقة انتهينا بالميكانيزم of action of the enzymes. ما تطرقنا للميكانيزم of action. اليوم راح نبدي إن شاء الله من الميكانيزم الخاص بالأنزيمات. mode of action قريناه طبعا وشرحناه إنه any action of the enzyme must be react or must be binding with the substrate by peptide bond. The mechanism of action depends on the decreasing of activation energy. Every action of the enzyme depends on the availability of active site. So the active site is the region or the place, as we said in the last lecture, it is the place that the substrate binds to the enzyme. Active sites, the definition of active site is depression or pocket on the surface of enzyme which the substrate molecule can fit. Substrate bind the enzyme causing or forming substrate enzyme complex. Reactions take place in the active site to convert the substrate molecule into what? Into product, releasing the enzyme without any changing in its properties or shape or formations. The product separates, leaving the enzyme free, unaltered, and unchanged. Now we will come to the mechanism of action, how this action is done, how the enzyme works on the substrate to produce a product. Enzymes increase reaction rates, which is it is work. What is the work of enzyme? It is work, or it is job is to accelerate or to increase the biochemical reaction rates. By what? By decreasing the activation energy, number one. Enzyme substrate by forming enzyme substrate interaction, by forming or formation or forming enzyme substrate complex. These mechanisms are done by two theories in the enzymology science. These theories are lock and key model or theory and induced fit model or theory. There are two models or two theories used in performance of mechanism of action of enzyme. There are lock and key model and induced fit model. Now we will take the steps or types of action, mechanism of action of enzyme point by point. Number one, as we said, the action of a mechanism of enzyme done by decreasing the activation energy. Before we talking or beginning to talk about the activation energy, we must know what is a transition state theory. Transition state theory, abbreviated as TST, is explain the reaction rate of elementary chemical reactions. Transition state theory is used primarily to understand qualitatively how chemical reactions take place. So, this theory is very important to know the chemical reactions steps, the chemical reactions mechanisms. So, the activation energy in chemistry can be defined as the minimum amount of energy or the lesser amount of energy, the smallest amount of energy that is required to activate atom. Atom is not active, it is inert. Activation energy is activate this atom or molecule. So, yeah, energy will activate the atom. Activation energy is minimum amount of this energy that is required to this atom to activate this atom or molecule to a condition in which they can undergo chemical transforming or physical transport. So, activation energy is the minimum or smallest or lesser amount of energy used to activate the inert atom, normal atom, to a condition in which they can undergo chemical transforming or physical transport to perform the biological or chemical test reaction. This is the definition of activation energy in chemistry. But it is definition in transition state theory. The activation energy is the difference in energy content between atoms or molecules in an activated or transition state configuration and the corresponding atoms and molecule in their initial configuration. In transition state, as we see, in transition state, activation energy is the difference between the transition state and initial state of between the atom or molecule in activated or transition configuration. Activation energy is usually represented by the symbol E capital A small. E refers to the word of energy, and small letter a, refers to the activation energy. In mathematical expression for such quantities as the reaction rate constant is measured in joule per mole. So when I answer you, what is the unit of determination of activation energy, you must answer. When I question you, you must answer that the unit of activation energy determination is joule per mole. When I ask you that the activation energy has a symbol in mathematical expression or not, you must answer that yes, the activation energy has a symbol E capital letter, A small letter in mathematical expression, and it is unit, determination unit is joules per mole or kilojoules per mole or kilocalories per mole. There are three expressions for the determination of activation energy. It can be determined by joule per mole or kilojoule per mole or kilocalories per mole. In this figure, the activation energy can be done by two reactants, reactant 1 and reactant 2. When there are two reactants to be react each other with each others by reducing the activation energy to produce a product. Potential, as we see in this figure, that's potential energy. By increasing the potential energy, the curve will be increased. And after forming the product in this, why the curve is increased? Because the reaction between the two reactants will be complete by using the energy of the reaction, by using the activation energy, a small amount of energy, the reaction between two reactants will reach the transition state, which is activated complex. And then the curve will fall down into the final state, which produced the product and release the enzyme outside the reaction without any changing. This differences between the transition state and the initial state of reactants is called activation energy. It's called the activation energy. The second type of the mechanism or second step of the mechanism is enzyme substrate intermediate. We said that the mechanism of action of the enzyme can be done, number one, by decreasing the activation energy, number two, enzyme substrate interactions. The enzyme and substrate form a reaction intermediate. Its formation has a lower activation energy than the reaction between reactant without a catalyst. When we have two reactants, reactant number one, reactant number two, when the two reactants reactions between by each others without using energy, without using enzyme or without using a catalyst, it is formation, it is activation energy will be high than using the enzyme as a catalyst. When we use catalyst, even if it is an enzyme or chemical catalyst, this activation energy will be decreased. If the reactions between two reactants is done in 10 minutes, for example, when we don't use the catalyst, it will done in 10 units. But when we add the catalyst into the reactions, into the chemical reaction container, this reaction will perform, or will be done in less than 10 minutes. It will be accelerate the time and accelerate the times of the other people who need the laboratory. Accelerate the time of reaction will also accelerate the time of the worker that work in the laboratory that use test for special analysis. The reactant, a simple light picture for this state or for this reaction, which done by lowering the activation energy, is forming intermediate. When we have two reactants, there will be formation of intermediate. Reactant number one plus enzyme equal intermediate. This intermediate will react with this intermediate react or plus reactant number two will form the product plus enzyme. So enzyme substrate intermediate step can be done by using two reactants with the enzyme. Reactant number one will act with the enzyme to form or equal intermediate. This intermediate will react with reactant number two to form product plus enzyme. The third step of mechanism or third type of mechanism is formation of enzyme substrate. Formation of enzyme substrate complex can be done by two types of models or two theories. Formation, the third step of enzyme mechanism, is formation of enzyme substrate complex, which is done by using two models or two theories in the enzymology science. The complete reaction of this step is substrate, as we see, substrate plus enzyme equal substrate enzyme complex, which lead to product enzyme complex, then equal product and enzyme. As we see in the three steps or three mechanisms of the enzyme mechanism, the enzyme releasing or release the chemical reaction without any changing or altering in its formation or its shape or its chemical or physical properties. It will be released free. It will be released free. This mechanism or this step of mechanism, as we see, we say, and as we see, depend on two models, lock and key model and induced fit model. Now we will coming to the lock and key model. The first mechanism or the first model discovered in the mechanism of action of the enzyme is lock and key model. The scientist Emil Fischer proposed this theory in 1894. When this theory is discovered, this theory or this model of mechanism of enzymes action is discovered in 1894 by Emil Fischer. This theory also can be named by Emil Fischer's theory. Emil Fischer's theory or Emil Fischer's model. Number two the specific action of an enzyme with a single substrate can be explained using this theory analogy. So, this theory, when is used, it can be used when there is only one substrate reaction or reactant reacts with the enzyme. When we have only one substrate reacts with the enzyme. 3. In this analogy, the log is the enzyme. We say this theory's name is log and key. Who is the log and who is the key? So the enzyme is the log and the substrate is the key. Enzyme is the log and the substrate is the key. Only the correctly sized key, which is the substrate, can be fits into the keyhole active site of the log of the enzyme, which is the enzyme. The enzyme is the log, substrate is the key. Only the correct key or correct substrate can be seed fitting or complete the active site of the locked correct. Only the corrected or correctly sized of the key can be used to seed in the active site, can be used to interaction with the active site of the enzyme, which is used as the lock for this key. Similar keys, larger keys, smaller keys, larger keys cannot be used, or incorrectly positioned teeth on keys, incorrectly shaped of keys, or incorrectly sized of keys cannot be fit into the lock. Only it is a specific substrate or it is a specific key is used to be fit or to be complementary for this lock. Only the correctly shaped key open the particular lock on the enzyme. يعني المفتاح الصحيح فقط هو هذا الأنزي أو هذا الميكانيزم أو هذا الثيوري أو النظرية بالميكانيزم of enzyme اسمه القفل والمفتاح. قلنا القفل يعتبر الأنزيم، المفتاح يعتبر السبستريت. السبستريت الخاص بهذا الأنزيم يقدر يتفاعل وياه، يقعد وياه فت. المفتاح مال الباب إذا ما يوالي من القفل، ما أعرف شو يسموه هذا المكان الموجود به القفل، خلينا نقول الموجود، إذا المفتاح سنون المفتاح ما يوالمه ما راح يفتح الباب. الأنزيم أيضا كذلك بمكان مثلا بها 4 سنون أو 5 سنون، يعني أشكال تشبه السنون خلينا نقول. إذا المفتاح ما بها التكملة مالتها مثل أيضا نفس الشيء مفتاح مال البيت مال أبواب البيت، إذا ما عنده نفس السنون المكملة لهاي الأماكن الموجودة بالقفل مستحيل يقدر يفتحها. الأنزيم أيضا إذا هذا السبستريت ما يقعد فت على الأكتف، شكل الأكتف سايت موجود عليها، ما راح يقدر يصير تفاعل بيناتهم، يسوي يعني interaction أو reactions between them. إذا كان هذا المفتاح كبير أو صغير أو غير ملائم بالشكل فما راح يصير هذا التفاعل. So this theory is very special or specific theory. The other substrates cannot react with the enzyme. Only it is specific substrates can be used by this enzyme to accelerate special or specific reactions. Both enzyme and substrate fit with each other correctly and tightly. The reaction between the substrate and the enzyme in this theory is very tight, is very tightly bonding or linkages. The bonds in this theory is very tight. Their shapes are complementary, as we say. The shape of the key or substrate must be complementary with each other. This bonding is very specific and cannot be easily broken. Is equalized into the lock and key mechanism. The lock and key model parts the lock and key model portrays as enzyme as conformationally rigid and able to bond only to substrate that exactly fit with the active site. الكهرباء انطفت، ما ادري اذا يسجل لو لا. الكهرباء انطفت، اذا يسجل شو اسمه لو لا. خوش يعني مع السلامه. العفو، طبعا قلت اتاكد انه اذا يسجل المحاضره اذا تتسجل لو لا. هسه النقطه السادسه، both enzyme and substrate fit with each other correctly and tightly. Their shapes are complementary، يعني المفتاح complementary with each مفتاح عن القفل، واحد مكمل للآخر. This binding is very specific and cannot be easily broken. So the lock and key theory differs from the other theory is in the reactions of the or bindings of the substrate with the enzyme cannot be easily broken, and one is complementary with each other. Their shape are complementary with each other. So it is called lock and key mechanism because it is equalized into the lock and key mechanism. In this figure, key is, as we mentioned, the key is like the substrate like the key, and the enzyme like the lock. Teeth of this key must be complement the shape of the active site in the enzyme. Correct fit will react. Incorrect substrate, no reaction. Cannot react with the enzyme. Cannot react. The other theory of the mechanism or other theory used to explain the mechanism of action or function of the enzyme is called induced fit theory. As we know that not all experimental evidence can be adequately explained by using the so-called rigid enzyme model assumed by the lock and key theory. Lock and key theory cannot be used in every mechanism, in all mechanisms of the enzyme's action. So, for this reason, a modification called induced fit theory can be proposed has been proposed. The induced fit theory is discovered by Cushland's induced fit hypothesis suggests in the presence of the substrate, the active site may be changed in order to fit the substrate's change. In lock and key substrate, there is no any changing in the shape of the active site of the enzyme. But in Cushland theory, the enzyme must be fit with the substrate shape. The active site may change in order to fit the substrate changes. So the enzyme initially have a binding configuration which attracts the substrate. Look at me, please. The active site consists of what? Active site, which we defined that it is a region or place on the surface of enzyme. But what is the formation of this active site? What is the nature of this active site that is on the surface of enzyme, which have two functions or two regions, also two sites, binding site and catalytic site. All these things depend on the structure of the active site. The structure of chemical formation of the active site is sequences of amino acids. These amino acids will bind with the substrate to by what? By the peptide bond. So, when the substrate has specific shape and the enzyme has another shape, differs from the shape of the substrate, the enzyme must change its configurations to be fit or to be complement for the shape of the substrate. When the substrate coming, the enzyme will attract it by changing its configurations and making its shape complementary to the shape of the substrate, making its shape to be similar or to be complement with the substrate shape to see it fit by on the active site or on this enzyme. The binding to the enzyme, the substrate disrupts the shape and causes it to assume a new configuration. The active site is then modeled into precise conformation. In this figure, as we see, the white color of molecules is active site. The substrate is colored by pink color. Induced fit theory suggests that the active site of the substrate, of the enzyme will change its conformation or will change its shape to be fit with the shape or to attract the substrate to react with each other by peptide bonds to forming substrate enzyme complex. So the reaction between substrate and the enzyme on the active site part of it is product the substrate enzyme complex. Substrate enzyme complex. As we mentioned before minutes, that the mechanism of action by forming substrate enzyme complex depends into two theories: lock and key theory and induced fit theory. So when the substrate react with the enzyme, it's causing or forming substrate enzyme complex. Then this complex will convert into product, releasing the enzyme freely. Releasing the enzyme freely. This substrate will be converted into product, releasing enzyme freely. This is another figure that explain or discuss this theory, induced fit theory. Substrate has two geometrical shapes, like curve, and this will be react with this active site. The active site has another shape, differs from the shape of substrate. But when the substrate react on the amino acids in the active sites, as we see, and forming peptide bonds, the enzyme changes its shape tightly as substrate bind, forming enzyme substrate complex. Substrate entering active site of enzyme, forming enzyme substrate complex. Enzyme substrate complex will convert into enzyme product complex. As you see these steps before reactions between substrate and the active site of the enzyme, then after reactions occurs between each other by peptide bond or peptide linkage, it will be converted into enzyme substrate complex. Enzyme substrate complex converted into enzyme product complex, equal product and enzyme. As we see, product leaving the active site of the enzyme, product leaving the active site of the enzyme, and the enzyme will be, these are products. The products leaving the active site of the enzyme. This enzyme is free, can be entered into another reactions, can be used in another biochemical reaction to accelerate the rate of its reactions. Now we completed the mechanism of actions of the enzymes. 3 steps of mechanisms, or 4 steps. 1, by decreasing activation energy, by decreasing by forming intermediate, by forming enzyme complex, enzyme substrate complex, by using two theories which are called Emil-Fischer theory or Kushlam theory, or lock and key theory and induced theory. And we will come into the specificity of enzymes. Enzymes, when they discovered the world cannot be, can't know that what is the enzyme and how it is how it is working, and what is the substrate. But in the in the coming years and the coming steps of the science life, the world or the scientists are discovered many informations, many properties about the enzyme. One of these properties of the enzymes is it is a specificity in the working, or in it is job, or it is working. Different types of enzyme especially, or specificity. Enzymes shows different degree of specificity grouped into 6 categories. So the specificity of enzymes can be divided or can be shows in different degrees, or can be grouped into 6 categories. Number 1, bond specificity. Bond specificity, if the bond is tightly formation, or it is loses the active site without any using any other chemical substances. It is not binding tightly to the enzyme. So the type of bond will act on the specificity of the enzyme. If it is tightly or weakly binding between the substrate and the active site of the enzyme. Number 2, in the bond specificity also there are many types of the bonding. In the peptide bond, we have several types of peptide bond. We will explain it later. Group specificity, each enzyme in this group or in this type of specificity, each enzyme can act on specific group. If like enzyme can be act on the hydrocarbons, enzymes can act on the lipids, enzymes can act on another molecules. Groups as a group, Substrate specificity. Each enzyme acts on a specific substrate. For example, sucrase act on the sucrose. Maltase acts on the maltose sugar. Urease acts on urea and others. Optical or stereospecificity. Enzymes act on group alpha optically actives on beta or levo or others. Geometrical specificity and cofactor specificity, if they have a specific geometrical shape or specific cofactors. A particular type of chemical bond or functional group. In general, there are 4 distinct types of specificity. As particular type of chemical bonds, there are 4 types of specificity. 1, absolute specificity. The enzyme will catalyze only 1 reactions. Group specificity, the enzyme will act only 1 molecules that have specific functional group, such as amino, phosphate, or metal groups. Amino groups, phosphate groups, metal group, and others. Stereochemical specificity, the enzyme will act on a particular steric or optical isomer. Linkage specificity, the enzyme will act on a particular type of chemical bond regardless of the rest of the molecular structure. So, linkage specificity can be described as the enzyme will act on particular type of chemical bond, act on particular type or a special type of the bond regardless of the rest of the molecular structure. طبعا ما اريد اتعبكم اكثر من هاي. لهنا ان شاء الله كملنا المحاضرة الثانية، بس كشيء يعني مراجعة بسيطة للمحاضرة الأولى. بدينا أول ما بدينا we start or begin the definition of biochemistry. What is the biochemistry? What is the science of biochemistry? Definition of it and the uses of biochemistry which can be used in agriculture, medicine, nutrition, clinical chemistry, pharmacology, and toxicology. Enzymology, as a part of or a branch of biochemistry, deals with the study of properties, activity, chemistry, nature, biochemistry, effect, and significance of the enzyme. بعدين اتطرقنا إلى الانزيمز. What are the enzymes? The definition of the enzyme are special type of protein, like all proteins made up of amino acid. The function of enzyme is determined by amino acid sequence, types of amino acid, that chain, and the chain of the shape of amino acid will affect the enzyme. Nature of the enzymes is found in all living cells and are made of protein and they are biological catalysts, speed up of reaction and unchanged by the reaction, and they are high molecular weights, can be denatured by salt, acid, or other reagents. They have molecular weight ranging from 10,000 to 2 millions, and the structure of enzyme consists of active site of the enzyme. Active site is the region or a place or depression on the surface of the enzyme. And the structure of enzyme is one of the structure of the protein. As we say, the protein has two types of structures: primary, secondary, tertiary, and quaternary. But the enzyme is the structure of it, which is a tertiary structure. And a change in the shape of a protein affects the shape of active site and the function of the enzyme. Active site has can be divided into further two types of sites or two places, for binding site and catalytic site. Binding site is responsible for choosing the substrate and binding it to the active site, but catalytic site works to performance the catalytic action of the enzyme. Many enzymes require another molecules or another compounds to do its job or to do its action correctly, more accurately, before the catalytic activity can be exerted. These compounds are called cofactors. This entire active complex is referred to as holoenzyme. A coenzyme is the protein portion of the enzyme plus the coenzyme or plus the cofactor. Even the cofactor is coenzyme, prosthetic group, or metal ion activator, is called holoenzyme. In this figure, we explain what is the holoenzyme. It consists of a coenzyme, which is a protein portion of enzyme, plus the cofactor, which contains the catalytic side, the coenzyme or metal ion, cation, or other types. coenzyme زائدا apo enzyme راح ينتج لنا هالو enzyme. Cofactor may be organic cation or organic molecules, usually vitamins. Organic groups are permanently bound to the enzyme, prosthetic group called prosthetic group. Cations, positively charged metal ion, or called activator, which are temporarily bind to the active site, not permanently, إنما بشكل مؤقت يرتبط ويا الactive site of the enzyme. And organic molecule as vitamins, coenzyme, which are not permanently bound also to the enzyme molecule but combine with enzyme substrate complex. So the coenzyme will bind with enzyme substrate complex and not with the enzyme directly. Type of cofactors, we have essential ions and coenzymes, activator ions, essential ion divided into activator ions, loosely bound, and metal ion or metalloenzymes, which is tightly bound. Coenzyme divided into prostatic group and co-substrates, which is loosely bound, and prostatic group tightly bound. Many enzymes require inorganic cation, metal activated, called metal activated, and metalloenzymes. Coenzymes, non-protein, organic, low molecular weight, dialysable substance associated with enzyme function known as coenzyme. They are often regarded to as second substrate or co-substrate. Coenzyme can be called second substrate or co-substrate since they have affinity with the enzyme comparable with the substrate. Type of coenzyme, we have two types or two groups, B-complex vitamin coenzymes and non-B-complex vitamin coenzymes. Coenzyme function and important energy product and transferring. Mode of action, as we say, that depend on the active site and the mechanism of action. There we have three types of mechanism or three steps of mechanisms: decreasing the activation energy, enzyme substrate intermediate, and enzyme substrate interactions. Formation of enzyme substrate complex depend or can be done by two types of theories or two types of models, lock and key models and induced fit models. Number one, decreasing the activation energy. Before we are talking about the activation, we talked about the transition state theory, which explains the reaction rate of elementary chemical reactions. Transition state theory is used primarily to understand qualitatively how chemical reactions take place. Activation energy in chemistry, or activation energy definition in chemistry, is the minimum amount of energy that is required to activate atoms or molecules to a condition in which they can undergo chemical transformation or physical transport. In transition state theory, the activation energy is the difference between the energy content, difference in energy content between atoms or molecules in the activated or transition state configuration and the corresponding atoms and molecules in their initial configuration. Activation energy is usually represented by symbol E capital I small in mathematical expression for such quantities as the reaction rate constant is measured in joule per mole or kilojoule per mole or kilocalorie per mole. This expression is done by this figure, that reactant 1 plus reactant 2 equal product. The activation energy is the differences between transition state energy and the initial state reactant and activated complex in the transition state. Enzyme substrate interaction. The enzyme and substrate form a reaction intermediate. Its formation has a lower activation energy than in the reaction between reactants, without catalyst. Presence of the catalyst can be accelerate the time of the reaction rate. Reactant number 1 plus enzyme equal intermediate. This intermediate plus reactant number 2 equal product plus enzyme. Formation of enzyme substrate complex done by two, depending on the two models, or done by two models. Lock and key model, induced fit model. Substrate plus enzymes equal substrate enzyme complex equal product enzyme complex. Substrate plus enzyme equal substrate enzyme complex equal product enzyme complex and lead to product plus enzyme. Lock and key model specific and rigid and tightly bounding model between the key and the lock. Incorrectly big or small lock cannot be used or cannot be done by this model. Both enzyme and substrate fit with each other correctly and tightly. Their shapes are complementary with each other, hence their binding is very specific and cannot be easily broken. This binding is very tight, is very specific, cannot be easily broken, is equalized into lock and key mechanism. This model conformationally rigid and able to bond only two substrates that exactly fit with the active site. If the substrate shape is not fit with the shape of active site, it cannot be done by this mechanism. This figure explained or discuss the definition or the action of the enzyme by lock and key theory. In this theory, not all experimental evidence can be adequately explained by using the so-called rigid enzyme model. Assume the lock and key theory. For this region, a modification called the induced fit theory has been proposed. Induced at fit theory proposed by Koshland's, which initially have a binding configuration which attracts the substrate, only binding to the enzyme. The substrate disrupts the shape and causes it to assume a new configuration. The active site is then modeled into the precise conformation. This figure explains the definition of the induced fit theory, and also this figure appears that the enzyme will change its shape according to the shape of the substrate. After the product appears or presents, the enzyme, the product will leave the enzyme freely. At the last, we talked about the specificity of the enzyme. There are six categories for this specificity: bound, group, substrate, optical or stereo specificity, geometrical, and cofactor specificity. A particular type of chemical bond function group. In general, there are four distinct types of specificity. This in chemical: absolute, group, stereochemical, and linkage specificity. إن شاء الله ألتقيكم الأسبوع القادم. إلى اللقاء.